Astaxanthin is a natural keto-carotenoid synthesized primarily by the unicellular microalga Haematococcus pluvialis. When exposed to harsh, intense ultraviolet radiation or nutrient stress, microalgae accumulate vast astaxanthin reserves as a cellular shield. This intense red, lipid-soluble pigment travels up marine food chains, concentrating in wild salmon, krill, and flamingos. Modern molecular nutrition has identified trans-astaxanthin as one of the most powerful natural antioxidants in nature. Unlike hydrophobic beta-carotene or hydrophilic vitamin C, astaxanthin features unique polar esterified end groups. These dual polar ends allow astaxanthin to span the entire lipid bilayer of human cellular and organelle membranes. By spanning the outer, inner, and hydrophobic core of cell membranes, it protects cell structures from oxidative attack. Research demonstrates astaxanthin neutralizes singlet oxygen with 6,000 times greater quenching capacity than ascorbic acid. It also exhibits 800 times greater lipid peroxidation inhibition than coenzyme Q10. Furthermore, astaxanthin never acts as a prooxidant under high oxidative stress, making it remarkably safe. To understand how astaxanthin protects human health, we must examine its impact on mitochondrial bioenergetics. Mitochondria generate cellular ATP energy through electron transport chains, producing reactive oxygen species as metabolic byproducts. Excess mitochondrial reactive oxygen species damage mitochondrial DNA, leading to progressive bioenergetic decay. Astaxanthin concentrates inside inner mitochondrial membranes, scavenging free radicals right at their production site. Protecting mitochondrial membranes preserves electron transport efficiency and prevents ATP synthesis decline. Clinical trials show astaxanthin supplementation increases skeletal muscle endurance by enhancing mitochondrial fatty acid oxidation. Let us examine the exact molecular mechanism by which astaxanthin protects human skin from solar photoaging. Solar ultraviolet radiation triggers cutaneous reactive oxygen species generation, activating matrix metalloproteinase enzymes. Activated matrix metalloproteinases degrade collagen fibers and elastin, causing deep wrinkle formation and loss of elasticity. Astaxanthin suppresses ultraviolet-induced MMP1 and MMP8 expression in dermal fibroblasts. By preserving dermal collagen architecture, astaxanthin improves skin moisture retention, elasticity, and fine line smoothness. In double-blind human clinical trials, 4 to 6 milligrams daily significantly reduced age spots and hyperpigmentation. Astaxanthin also exhibits potent vascular anti-inflammatory activity by downregulating nuclear factor kappa B. It suppresses pro-inflammatory cytokines like interleukin-6 and tumor necrosis factor alpha in vascular endothelial cells. Suppressing vascular inflammation improves systemic arterial flexibility and lowers resting blood pressure. In neuronal cells, astaxanthin crosses the blood-brain barrier, protecting hippocampal neurons from neurotoxic oxidation. By suppressing microglial neuroinflammation, astaxanthin supports cognitive processing speed and working memory. Astaxanthin also supports ocular health by accumulating in retinal capillary membranes and ciliary bodies. It enhances choroidal blood flow, reducing digital eye fatigue and age-related macular oxidative damage. Let us analyze how astaxanthin modulates cellular autophagy pathways. Autophagy is the lysosomal degradation process that clears damaged cytosolic proteins and dysfunctional mitochondria. Astaxanthin promotes autophagic clearance by upregulating AMP-activated protein kinase and suppressing mTOR. Clearing defective mitochondria prevents cytosolic leakage of inflammatory mitochondrial DNA. This autophagic renewal mitigates chronic tissue inflammation, supporting long-term organ vitality. To maximize oral absorption, consume natural astaxanthin alongside a meal containing healthy dietary lipids. Dietary fats stimulate bile acid secretion, forming lipid micellar complexes that carry hydrophobic astaxanthin across gut mucosa. Dividing daily intake into 4 to 8 milligram doses maintains steady plasma concentrations and tissue saturation. Combining astaxanthin with physical exercise, polyphenol-rich foods, and restorative sleep unlocks optimal longevity. Let us examine the pharmacokinetic profile and bioavailability of microalgae-derived astaxanthin. Natural astaxanthin from Mitochondria pluvialis is esterified with fatty acids, granting far superior stability over synthetic forms. Following oral intake, peak plasma concentration occurs within 8 to 12 hours. Astaxanthin is incorporated into chylomicrons, transported via lipases, and distributed into cell membrane phospholipid bilayers. Because astaxanthin does not interfere with blood clotting or liver cytochrome enzymes, it features an exceptional safety profile. Regular supplementation ensures steady tissue accumulation in cardiac, neuronal, dermal, and ocular tissues. Research continues to confirm astaxanthin as a cornerstone nutrient for cellular longevity and bioenergetic defense. Let us explore how astaxanthin protects vascular endothelial cell mitochondria during oxidative stress. Endothelial cells lining blood vessels depend on mitochondrial health to produce nitric oxide gas. Astaxanthin prevents peroxynitrite radical formation in endothelial cells, preserving nitric oxide bioavailability. Preserving nitric oxide ensures continuous microvascular vasodilation and optimal organ blood perfusion. In liver tissue, astaxanthin downregulates sterol regulatory element binding protein 1C. Dovnegulatin sreb, se subpresses de novo lipogenesis, preventing hepatic trigliceride accumulation. In renal tissue, astaxanthin suppresses mesangial cell extracellular matrix expansion. Suppressing mesangial expansion protects renal glomeruli from hyperglycemia-induced oxidative fibrosis. In pancreatic tissue, astaxanthin protects insulin-producing beta cells from oxidative apoptosis. Preserving pancreatic beta cell mass supports long-term endogenous glycemic control. Furthermore, astaxanthin upregulates nuclear factor erythroid 2-related factor 2, or NRF2. Activated NRF2 enters cell nuclei, driving endogenous heme oxygenase 1 and superoxide dismutase transcription. Boosting endogenous antioxidant enzymes provides comprehensive protection against environmental oxidative stressors. In joint tissue, astaxanthin inhibits cartilage-degrading matrix metalloproteinase 13. Inhibiting MMP 13 preserves articular cartilage integrity, supporting joint mobility and reducing exercise soreness. In summary, natural astaxanthin is a unique membrane-spanning carotenoid that empowers cellular resilience. By protecting mitochondrial membranes, reducing photoaging, and boosting cellular energy, astaxanthin supports lifelong health. Integrating this marine nutrient into your daily routine provides evidence-based bioenergetic stamina. Let us examine the exact bioenergetic interaction between astaxanthin and complex 4 of the mitochondrial respiratory chain. Complex 4, or cytochrome C oxidase, transfers electrons to oxygen molecules during oxidative phosphorylation. Astaxanthin prevents lipid peroxidation in cardiolipid molecules that anchor complex 4 to inner mitochondrial membranes. Preserving cardiolipin structural integrity maintains optimal electron flux and prevents cytosolic cytochrome C leakage. In skeletal muscle myocytes, astaxanthin upregulates peroxisome proliferator activated receptor gamma coactivator 1-alpha. Elevated PGC 1-alpha stimulates mitochondrial biogenesis, increasing muscle endurance and oxidative capacity during aerobic exercise. Astaxanthin also suppresses exercise-induced plasma creatine kinase elevation, accelerating muscle tissue recovery. In cardiac myocytes, astaxanthin reduces ischemia-reperfusion ROS generation, preserving myocardial contractility. In brain astrocytes, astaxanthin enhances brain-derived neurotrophic factor, or BDNF, transcription. Elevated BDNF promotes synaptic plasticity, dendritic spine density, and long-term memory formation. Furthermore, astaxanthin inhibits aldose reductase enzymes in lens epithelial tissue, preventing osmotic cataract formation. In intestinal mucosa, astaxanthin enhances tight junction proteins like claudin-1 and agglutinin. Strengthening gut mucosal barrier function prevents bacterial endotoxin translocation and systemic metabolic endotoxemia. Astaxanthin also downregulates inducible nitric oxide synthase, or INOS, in activated macrophages. Suppressing INOS prevents excessive peroxynitrite synthesis, dampening acute inflammatory tissue destruction. In adipose tissue, astaxanthin upregulates adiponectin secretion, enhancing peripheral insulin sensitivity. Elevated adiponectin stimulates fatty acid beta oxidation in liver and skeletal muscle tissues. In renal podocytes, astaxanthin suppresses advanced glycation end-product receptor, or RAGE, signaling. Suppressing RAGE signaling protects kidney glomeruli from chronic diabetic microvascular filtration damage. Astaxanthin also enhances cutaneous skin-blood microcirculation, supplying oxygen and nutrients to epidermal layers. Improved dermal microcirculation accelerates natural wound healing and cellular skin turnover. In endothelial cells, astaxanthin upregulates vascular endothelial growth factor receptor 2 signaling. Controlled VEGFR2 activation supports physiological angiogenesis without promoting pathological microvascular leakage. These comprehensive multi-organ protective actions solidify astaxanthin as a primary longevity compound. In retinal pigment epithelial cells, astaxanthin upregulates endogenous glutathione peroxidase and catalase enzymes. Upregulating retinal antioxidant enzymes shields sensitive photoreceptor cells from high-energy blue light oxidation. In skeletal muscle mitochondria, astaxanthin preserves uncoupling protein 3 expression during prolonged physical stress. Preserving uncoupling protein 3 prevents mitochondrial hyperpolarization and excess superoxide leakage. In dermal microvessels, astaxanthin inhibits endothelial cell adhesion molecule 1 expression. Inhibiting adhesion molecules prevents chronic cutaneous microvascular inflammation and tissue degradation. Disclaimer: this video is for educational purposes only and does not constitute medical advice. Always consult a qualified healthcare professional regarding any medical condition or dietary changes. Thank you for watching Calorierecipes.